Welcome to our video lecture on photosynthesis. Our essential understandings for today: photosynthesis uses the energy in sunlight to produce chemical energy needed for life. That's of course going to be ATP that comes from the glucose that we make in photosynthesis, and then we convert it during respiration. We're also going to look a little bit at the extra HL details, light energy converted into chemical energy. And our objectives for today: we're going to review plant and plant cell structure. We're going to describe the structure and the function of chloroplasts. We're going to explain the two main stages of photosynthesis, the light-dependent reactions and then the light-independent reactions. We're going to explain the absorption and action spectrum of chlorophylls. What kind of light does chlorophyll even like? We're going to explain Calvin's Lollipop Investigation, the super cool experiment that helped Melvin and Calvin figure out how Calvin's cycle even happens. We're going to describe the factors that affect the rate of photosynthesis, and then we're going to have a super brief comment on how photosynthesis led to a change in oxygen levels in Earth's environment. So super quick look here at plants. So of course plants have roots, they have stems, they have leaves. We're going to focus in photosynthesis on these leaves. So this picture over here is a cross section through a leaf. On the very top, there's this waxy layer called the cuticle that helps to prevent dehydration of the leaf tissues. We've got the upper epidermis. It's like the skin, kind of, sort of the leaf. And then right under that, we have this Palisade Mesophyll. This is where the great majority of photosynthesis is occurring. And so we will often talk about these Palisade Mesophyll cells, oodles and oodles of chloroplasts in these guys. So here is a Palisade Mesophyll cell. Notice all of those beautiful chloroplasts. This is what we're going to be focusing on today. So we're going to focus in on chloroplast structure. Chloroplasts have double membranes. This is evidence for endosymbiosis. So we've got this outer membrane and inner membrane. We're pretty sure that inner membrane came from the original bacteria that was engulfed. This second outer membrane is the membrane that was formed by the vesicle that was formed during endocytosis. Inside the double membrane, we have the stroma. The stroma is this juicy part of the chloroplast that's outside of these stacks of membrane pancake-looking things called the thylakoid. So we have thylakoid here, lots and lots of thylakoid, again, that kind of look like pancakes. A stack of these pancake thylakoid membranes is called a granum. Grana is plural, so lots of granums is a grana. Lamella are going to connect the grana to each other. So we're going to focus a lot on thylakoid and stroma when we talk about the reactions that occur in photosynthesis. The thylakoid has the electron transport chain, Atp synthase, and the chlorophylls. So chlorophylls that are part of our photosystems are embedded in all of these pancakes, these thylakoid membranes. Also, ATP synthase is embedded in these membranes. So the first stuff that we're going to talk about, the light-dependent reactions, happen along those thylakoid membranes. The light-independent reactions happen in the stroma. So we'll talk a lot about thylakoid first, and then we'll look at the stroma next. Here's a micrograph of a chloroplast. So just looking at this structure, but applying it to an actual chloroplast, we have double membrane here. It's hard to tell that it's a double membrane because they're so smack close to each other, but this is double membrane. Here's going to be some cell wall. Here's cytoplasm of the plant cell. These are those grana, those stacks of pancakes made of thylakoid membrane. So here's our thylakoid, and then in between those thylakoid membrane stacks are the stroma, and then these are our lamella that are connecting those stacks. Thylakoid, this is where the light-dependent reactions occur. We'll talk about that a lot. Stroma, this is where our light-independent reactions occur. We also call the light-independent reactions Calvin's cycle. All right, so photosynthesis, we're going to take some carbon dioxide and some water. We're going to use energy from light. We're going to use these amazing chlorophyll molecules, and we're going to be able to form glucose, oxygen, and water. And by we, I mean plants, not actually us humans. All of this is going to occur in the chloroplast. So here's our chloroplast. We're going to start with the light reactions. We're going to talk about light reactions first. Both of these can happen at the same time, but we just like to talk about light reactions first. We're going to use water in the light reactions. We're going to make a little bit of ATP, a little bit of NADPH. We're going to use that ATP and that NADPH that we make in the light reactions, in the light independent reactions, also called Calvin cycle, light independent. So clearly light dependent, we need that light to make these guys occur. The light independent reactions, Calvin cycle, no light required. But now we do need that CO2. So we're going to use CO2 in Calvin cycle. We're going to use the water in the light reactions. In the light reactions, we make some ATP and some NADPH. In Calvin cycle, we make some sugar. What's kind of cool about photosynthesis and respiration is that they're almost opposites of each other. So in the chloroplast and photosynthesis, we're going to make some glucose. Oxygen gas is a waste product. In the mitochondria, in respiration, we're going to use oxygen and glucose to make lots of chemical energy in the form of ATP. The waste products of cellular respiration are water and carbon dioxide, which is used by the chloroplast with some energy from the sunshine to make oxygen gas and glucose. That glucose and the oxygen are used, and it just keeps going around and around this beautiful circle. Sometimes we get a little bit too simplified and we're like, these are plants and these are animals. That's wrong. Remember that plants also have mitochondria. So don't forget that those plants are not just chloroplast filled, but they also have their own mitochondria. Here's a quick overview of photosynthesis. So we've got our light dependent reactions. This needs some sunshine. Happens on the thylakoid membranes. We're going to take that water, we're going to split it up, we're going to make some oxygen. It's a waste product. We have also hydrogen and electrons. These are used to reduce NADP plus into NADPH. We're also going to make some ATP with that ATP synthase. These guys, NADPH and ATP, are used in the light independent reactions, which is also called the Calvin cycle. This happens in the stroma of the chloroplast. We're going to use that carbon dioxide along with the energy and the electrons and the protons from NADPH and ATP to make some organic compounds, basically glucose. I like to talk about the light dependent reactions first, but definitely hear me say light dependent and Calvin cycle can happen at the same time. We just have to start somewhere. I like to start here. So light dependent reactions, photolysis was one of the first things that happens. All of this is happening along the thylakoid membrane. So here is our thylakoid membrane. So imagine this goes around into that pancake, right? So there's our little pancake. This is the stroma. This is outside the pancake. Here's the inside of the pancake. We can call it the thylakoid lumen. Lumen means the inside of anything. Sometimes we just call it the thylakoid space, but we're basically inside this little pancake. Photolysis is going to occur. We're going to take some water. We're going to use light energy, that's the photo part, to split, that's the lysis part, this water. We're going to get some oxygen gas, we're going to get some hydrogen ions and some electrons. Oxygen gas is a waste product. It is going to diffuse out of the thylakoid, out of the stroma, out of the chloroplast, and eventually out of the whole entire plant. Light energy is going to zap this guy called photosystem 2. Photosystem 2. So we're going to zap some energy into those electrons, these electrons that are passed onto photosystem 2. The high energy electrons, they're high energy because they absorbed energy from the sun. They're going to jump along this electron transport chain. Energy from the electrons jumping is going to be transferred to these proteins of the electron transport chain. That energy is going to be used by those proteins to pump hydrogen ions, also called protons, also called H pluses, all the same thing. We're going to pump those protons into the lumen of the thylakoid membranes. So that's going to lead to a high concentration gradient of these hydrogen ions. We're going to have a bunch of hydrogen ions in here because those electrons jump, 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 give energy to the electron transport chain. That energy is used to pump the hydrogen ions into the lumen. High concentration gradient, proton gradient, high concentration gradient of these protons, these hydrogen ions inside the thylakoid lumen, or the space of the thylakoid. Of course, we know that the universe is not stable when we have a bunch of things all smashed together like this. Those hydrogen ions need to diffuse away from each other. Their only path, though, out of the thylakoid space is through ATP synthase. So those hydrogen ions are going to move through ATP synthase in order to diffuse back to the stroma to get to equilibrium. Interestingly, ATP synthase can use that energy, that kinetic energy of those hydrogen ions moving through it to add an inorganic phosphate group to ADP to make ATP, just like ATP synthase in our mitochondria. So hydrogen ions flow through ATP synthase. ATP synthase can take a little bit of phosphate, some ADP, and make a little bit of ATP. This is not the ATP that the cell uses for everything. This is ATP that it uses only to make glucose. So one more time, we have those electrons. Those electrons are going to get zapped on photosystem 2. Those electrons come from the photolysis of water. Electrons jump, 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 jump. Energy is transferred to the proteins of the electron transport chain. That energy is used to pump these protons into the lumen, that thylakoid space. Those hydrogen ions make a crazy high concentration gradient. They're going to diffuse through ATP synthase, facilitated diffusion. That is going to allow ATP synthase to have the energy that it needs to combine ADP and an inorganic phosphate group to make ATP. This is called chemiosmosis. Chemiosmosis. So there's a little piece that we haven't talked about that yet. This guy right here. Photosystem 2 we talk about first. It's where the water gets split and the electrons get zapped. But then those electrons that now have lost all that energy, they are passed from the electron transport chain onto photosystem 1. Photosystem 1 re-zaps those electrons, so they're high energy again. So those electrons that lost all that energy, now they have lots of energy again because they get re-excited by sunlight energy on photosystem 1. And then they get passed to NADP+, which is reduced to NADPH. Remember that reduction is the gain of electrons. Leo says GER, gain electrons is reduction. So they gain some electrons and some protons and turn into NADPH. That NADPH is used along with that ATP to make some glucose. So I have mentioned these photosystems. Photosystems are where we have chlorophyll molecules, and chlorophyll molecules are super cool. When they absorb energy from the sun, they pop out some electrons. And so those electrons can jump from one chlorophyll to the next, because this guy will lose an electron and get passed to the next guy, which will lose an electron that gets passed to the next guy. So when we're talking about chloroplasts, chlorophyll in our photosystems, chlorophyll is a plant pigment that's really good at absorbing light energy. It's not the only one, though. We also have xanthophylls and carotenoids that can also absorb light energy. This graph is showing us the different wavelengths, colors of light that are absorbed by these different pigments. What's kind of crazy, notice my friends, we've got lots of blues here, peaks around the blues. We have some more peaks over here in the oranges. Notice what color is least absorbed. Green. Plants look green to our eyeballs because that's one of the colors that is not well absorbed by the chlorophylls in the photosystems. This is the color that gets reflected back to our eyeballs. So because plants don't like green, they look green. So before we move on to Calvin's cycle of the light independent reactions, we're going to review the light dependent reactions one more time. That chlorophyll that we just talked about, xanthophylls and carotenoids, those pigments are here on photosystem 2, photosystem 1. The first thing that's going to happen is that photolysis. lysis of water. Water is going to get split apart into oxygen gas that is removed as a waste product. We have those hydrogen ions, also called protons, and then we've got those electrons. Our electrons are going to get zapped by sun energy, or whatever kind of light you've got. And those electrons are going to gain that energy. They're going to use that energy to jump along the electron transport chain. The electrons pass their energy to the proteins of the electron transport chain. Those proteins are going to use that energy to pump hydrogen ions into the inside of the thylakoid. Sometimes we call it the lumen, sometimes we just call it the thylakoid space. That's going to lead to this high concentration gradient of hydrogen ions. We call it sometimes a proton gradient, high concentration gradient of protons in the inside of the thylakoid membrane. That proton gradient is going to lead to chemiosmosis, because this high concentration gradient is not stable. Those hydrogen ions are going to diffuse through ATP synthase. ATP synthase also embedded in that thylakoid membrane. The hydrogen ions flow through ATP synthase. That gives energy to this enzyme complex, and it is going to glue together some ADP and some inorganic phosphate to make some ATP. That ATP is used in the Calvin cycle, which is also called the light-independent reactions. We're going to go back up to those electrons, those pesky electrons right here. So those electrons on the electron transport chain, they're low energy. They gave their energy away to the electron transport chain so they could pump those hydrogens, right? So those electrons at the end of the electron transport chain are going to get passed to photosystem 1, and they're going to get more light energy. That light energy is going to once again zap those electrons to high energy, and those electrons are then going to be passed to NADP+, which is turned into NADPH. We're going to reduce NADP+ into NADPH. This is also used in the Calvin cycle. Everything we've been talking about so far is happening here in the thylakoid membranes, in these pancakes. The electron transport chain, photosystem 2, photosystem 1, ATP synthase, these are all embedded in the membrane that is the thylakoid. The reason that we have those little pancakes is so that we have lots and lots of surface area for lots and lots of photosystem 1, photosystem 2, electron transport chain, ATP synthase. It also is so that we have this little bit of space so that we can fill them with those hydrogen ions super fast and build up that high concentration pretty quickly. That's all the light-dependent reactions happening here. Now we're going to move out of the thylakoid into the stroma and talk about Calvin cycle. So what we talked about before is all happening here along the thylakoid membranes. Now we're going to move out to the stroma and the Calvin cycle. And here is Calvin cycle, also called the light-independent reactions because no light is required. This does not happen only in the dark. It definitely happens at the same time as the light-dependent reactions. It just doesn't need light. It does, however, need the products, that ATP and NADPH that was made during the light-dependent reactions. So what happens in Calvin cycle? We're going to take that carbon dioxide that diffuses into the plant. It is going to get bound to RUBP, ribulose biphosphate, also called RUBP, this molecule right here. The enzyme that makes this happen is called Rubisco, most common enzyme on the planet. So CO2 plus RUBP is going to make 6 molecules of this stuff called glycerate 3-phosphate. We call it GP for short. RUBP plus CO2 bound together by this enzyme Rubisco is going to make 6 molecules of GP, glycerate 3-phosphate. We call that carbon fixation because to fix something means to glue it together, and we're literally gluing the carbon to this RUBP stuff. 6 molecules of GP, glycerate 3-phosphate. We're going to use the ATP and the NADPH that we made in the light-dependent reactions on the thylakoid membranes. We're going to use this stuff to now reduce glycerate 3-phosphate to triose phosphate. Remember that reduction is the gaining of electrons and protons. Leo says Ger. So these Electrons and protons that used to be on NADPH are going to get chunked onto GP, which is going to turn GP into TP. So that's why we call it reduction, because GP gains some electrons and some protons and turns into TP. We have 6 molecules of TP, triose phosphate. Here's the weird thing. One of those TPs is going to exit Calvin's cycle, and it's basically half a glucose molecule. We have half a glucose. The other 5 triose phosphates are regenerated back into RUBP so that we can do the whole thing all over again. If we used up all our RUBP, we could only do this one time, and then you'd have half a glucose, and that was the end. So instead, we're going to make that RUBP again. That RUBP is going to be used, bind together with some CO2 by the enzyme Rubisco. We're going to make some more GP. We're going to turn that into some more TP. We'll get another half a glucose, and 1/2 plus 1/2 equals a whole glucose molecule. 5 of the TP that were made this time get turned back into RUBP. We're going to combine that RUBP with CO2 using the enzyme Rubisco. That's going to form 6 molecules of glycerate 3-phosphate GP. That GP is going to be reduced to TP, triose phosphate. We have 6 of them. 1 of them exits the cycle. The other 5 are regenerated back into RUBP. And it keeps going around and around, which is why we call it a cycle. This cycle was figured out by this guy named Calvin. His name is actually Melvin Calvin. I don't know why anyone would do that to their child, but someone did. Anyhow, we call it the Lollipop Investigation because it kind of looks like a lollipop, right? So what Calvin did was he put some algae into this glass part of the lollipop. The algae was happily doing some photosynthesis. He started adding a little bit of carbon dioxide that was radioactive. So he had carbon dioxide that had carbon-14. That's a radioactive version of carbon. So this radioactive CO2 was being added into the algae through photosynthesis. Calvin would drip out some of the stuff that was made by the algae in the lollipop, and then he put it basically on X-ray film to figure out what molecules were radioactive. The first thing that was radioactive was GP. And that told him that the first thing made in the light-independent reactions was glycerate 3-phosphate GP. After a little while, the next thing that he saw pop up was triose phosphate. So he knew that the second thing made in Calvin's cycle, we call it Calvin because he's Calvin, the second thing made in the cycle was TP. And then he started to notice that there was RUBP, like this is lots of that stuff, RUBP was also radioactive. That allowed him to figure out that that RUBP was being regenerated and then used to make more GP. So by adding carbon-14 in carbon dioxide to the algae, Calvin was able to figure out the different steps in Calvin's cycle. And that's why we call it Calvin's cycle. As with many of our biological processes, we can change the rate of photosynthesis, right? And there are lots of different things that can change how fast photosynthesis occurs. Light is one of them, of course. No light, no photosynthesis. The reason is because photosystem 2, photosystem 1, they need to use that light energy to make things happen in the light-dependent reactions. Little bit of light, more photosynthesis, little bit more light, more photosynthesis. But at some point we're going to saturate, and more light won't make a difference to the rate of photosynthesis. This is because photosystem 2 and photosystem 1 are saturated. We have plateaued how much energy can be absorbed by these guys. It doesn't matter if there's more light. These guys cannot work any faster. And so that rate is going to plateau because these enzyme complexes, these protein complexes are saturated. They have as much light as they can possibly absorb. CO2 has a similar graph, and the reason is Rubisco. So Rubisco is that enzyme that binds together carbon dioxide and RUBP during the carbon fixation part of Calvin's cycle. little bit of CO2, a little bit of photosynthesis, more CO2, more photosynthesis, until we get to this point where it doesn't matter that we have more CO2. More CO2 is not going to change the rate of photosynthesis. The reason is because Rubisco is now saturated. And we're going to have a plateau on our graph. Rubisco is working just as fast as it can. More CO2 is not going to make a lick of difference. Final guy is temperature, and this looks a lot like our other temperature enzyme graphs. So we have that optimal temperature. This is where Rubisco and PS2, PS1 are working as fast as they can. Lower temperature means that we just don't have enough collisions happening with enough energy to get things to go. Too much temperature, and now we're starting to denature those proteins, Rubisco, PS2, PS1. So one little comment on oxygen levels on Earth before we call this lecture done. So a long time ago, there was no oxygen in the atmosphere or in the oceans. Earth had no oxygen. But then this weird photosynthesis mutation popped up, and it was really good. And so lots of things that could do photosynthesis had lots of babies, and they started making some oxygen. For a while, it was stuck in the oceans, and then it got stuck to rocks. But then it started to accumulate in the atmosphere. And so we had more oxygen popping up, which allowed lots of aerobic living things to evolve and become cool like us. Interestingly, though, that oxygen in the environment also killed off a bunch of bacteria that were sensitive to oxygen. So we had our first big mass extinction because oxygen. Crazy sauce, right? And we did it. We reviewed plant structure, plant cell structure. We talked about chloroplasts. Remember that they've got double membrane. They have those pancakes that are called thylakoid, and outside the thylakoid is the stroma. Two main stages of photosynthesis. The first stage, light-dependent reactions, happens on the thylakoid. The other one happens in the stroma. Calvin cycle, also called the light-independent reactions. In that first stage, we make some ATP and some NADPH, and that's used in the second stage to make some glucose. We talked about that absorption and action spectrum of chlorophylls. They don't like green. They act a whole lot better with reds and purples. We explained Calvin's Lollipop Investigation, where he added radioactive CO2, carbon-14 in the CO2, and he saw that GP got made first and then TP got made next, and then it turned back into RUBP, ribulose biphosphate. We talked about those factors that affect the rate of photosynthesis: temperature, just like all the other enzymes, CO2 concentrations, and light intensity. And we wrapped up with a comment on how oxygen killed things when it started to form because of photosynthesis. Good job today.